We have completed the first part of the transport across cell membrane, that is the passive transport. Now come to the active transport. Active transport processes, they utilize the energy by hydrolysis of ATP. Okay, so there are mostly two types of active transport. One is the primary active transport, another is the secondary active transport. In the primary active transport, the energy is derived directly from the breakdown of ATP. What are the examples of primary active transport? Most important example is a pump, that is sodium potassium ATPase pump. Okay, other examples, so many these transporters are mostly called the pump because they are pumping against the concentration or the electrical gradient. Other examples, it may be H plus K plus ATPase pump in the gastric mucosa and in the renal tubules. It may be the calcium ATPase pumps that pumps out the calcium out of cells. H plus ATPases acidify many intracellular organelles, namely the lysosomes. So there are certain examples of primary active transport. Now in this video we will specially discuss about the most important pump of our body, that is sodium potassium ATPase pump. Now sodium potassium ATPase pump, it uses the energy to move 3 sodium out of the cell. We know from our previous video, that is the body fluid distribution and the ionic concentration, we know that extracellularly the most higher concentrated ion is the sodium ion. So extracellular sodium concentration is high and intracellular potassium concentration is high. Okay, so here the sodium is moving from already the sodium is high at the extracellular space, so instead of this fact it is moving against the concentration gradient. So what should be the normal tendency of the sodium? To move from outside extracellular to intracellular, but as the concentration is higher at the outside of the cell. But here with the help of this pump, the sodium is moving, 3 sodium ions are moving out of the cell, so against the concentration gradient. And we will also see that intracellularly it is negative and extracellularly it is positive. So when the normal tendency will be this intracellular negativity will try to retain this positive sodium ion inside. Okay, but against the electrical gradient also the sodium is moving outside. Okay, so the sodium is moving against its concentration and the electrical gradient. Now with the help of this pump, and another function this pump does, that it moves two potassium particles, it moves two potassium ions from outside to inside. We know that at the intracellular part the potassium is already high, so concentration of potassium is high, it is inside. So normal tendency of potassium will be it should move from inside to outside, but against this concentration gradient 2 potassium ions are coming inside. Okay, so what is the function of sodium potassium ATPase pump? It utilizes energy to move 3 sodium ions from inside to outside of the cell and move 2 potassium ions from outside to inside the cell. But in case of potassium, the movement is against the concentration gradient but along the electrical gradient, because inside it is negative, it will obviously attract the positively charged ion. Okay, so we can say that the coupling ratio of this sodium potassium ATPase pump, it is 3:2, that is 3 sodium outside and 2 potassium is going inside, and it is an electrogenic pump, which means it moves 3 positive charges out of the cell for each 2 that it moves in. Okay, that means the more number of positive ions are going outside and only 2 positive ions are coming inside. So we can call it as an electrogenic pump. Now if we want to see the structure of the sodium potassium ATPase pump, you will see that it is actually heterodimer. It has 2 subunits. Both of the subunits are different. It has 2 subunits. We can see one is the alpha subunit, another is the beta subunit. Alpha subunit has the intracellular and extracellular binding sites. The intracellular portion of alpha subunit, it has a sodium binding site, that is 1. It has a phosphorylation site, that is 4, and it is an ATP binding site, that is 3. And the extracellular side of this alpha subunit, it has potassium binding site, that is 2, and the Waben binding sites, that is 3. Whereas the beta subunit is a glycoprotein which has 3 extracellular glycosylation sites. Now, how this actually functions, we will see it in the next slide. How this sodium potassium ATPase pump acts: when the sodium ion or sodium, it binds to the intracellular binding site of the alpha subunit, ATP also binds to the phosphorylation site, and ATP is converted to ADP, with the phosphate being transferred to aspartate 376 and the phosphorylation at the phosphorylation site. So we have seen that intracellularly we had the one is the sodium binding site, one is the ATP binding site, another was the phosphorylation site. So when sodium binds, so ATP also binds and it is converted to ADP, and the phosphate will be transferred to the aspartate 376 at the phosphorylation site. So that will cause a configurational change. Okay, so this causes a change in the configuration of the protein, and it will cause the extruding sodium ions into the extracellular fluid. So it is move, the some configurational change will happen, and so that the channel is now open on the extracellular side and the sodium ions will go out. Okay, now then what will happen? Then in the extracellular space, what happens? The potassium will bind extracellularly at its binding site, and there will be dephosphorylation of the alpha subunit, which returns to its previous conformation and releasing the potassium inside the cell. Okay, so first attachment of sodium, then hydrolysis of ATP, then the phosphorylation, then configurational changes or conformational changes, and it will extrude 3 sodium ions outside. And then the potassium ion will attach, there will be dephosphorylation of alpha subunit, it will return to its previous conformation and it will release the 2 potassium inside the cytoplasm. So in this slide, by animation, we can see, so first the sodium ions are attaching, then the ATP will be hydrolyzed, the phosphate will be transferred, and there will be phosphorylation and the configurational changes. The sodium ions will move outside, then the potassium ion will come and they will attach, bind to their side, there will be dephosphorylation, phosphate is going out. Then again it will come to the original conformation and potassium is released inside. Now some important information regarding the regulation of sodium potassium pump. The active transport of sodium potassium pump is one of the major energy using processes in the body. On an average, it accounts for about 24% of the energy utilized by cells, and in neurons it accounts for around 70%. Now there are certain hormones which enhances the activity of this sodium potassium ATPase pump. One hormone is most importantly, that is a thyroid hormone. Another is the aldosterone. Another is insulin. They will increase the activity of this sodium potassium ATPase pump. On the other hand, cardiac glycosides like digitalis or digoxin, it will inhibit the activity of the pump, and OABN also, it will also inhibit the activity of this pump. Dopamine in the kidney, it inhibits the sodium potassium ATPase pump by phosphorylating it and causing the natriuresis. So these are the regulators of the sodium potassium ATPase pump inside our body. Other than sodium potassium ATPase pump, some other common ATPases are calcium ATPases, H plus clay plus ATPase, and H plus ATPase. They are classified into P type or phosphorylation type, V type or vacuolar type, F type, energy coupling factor type, and ABC transporters. Among them, CFTR or cystic fibrosis transmembrane regulator protein. It is involved in genesis of cystic fibrosis. And MDR1 protein, that is multi-drug resistant 1 protein, that pumps anti-cancer drug out of cancer cells, that causes drug resistance in treatment of cancer. The second part of active transport is type of active transport is the secondary active transport. We have already gone through the primary active transport. The example was sodium potassium ATPase pump. Now come to the second type, that is the secondary active transport. In primary active transport, I told you that it utilizes the energy by hydrolysis of ATP directly. Okay, so it directly uses the ATP, breakdown of ATP, and takes the energy for the movement of ions against the concentration or electrical gradient. Here actually in secondary active transport, it is an active transport. It requires the energy, but not directly at the surface of cell membrane where this transport process is happening. The best example you will see in case of renal tubular cells or the intestinal cells. So here actually what happens, these luminal cells, the cells in the intestinal lumen or the renal tubular lumen, they have got two surfaces. We can see here one surface that faces the lumen, that is a luminar surface, another surface that is opposite to the lumen, which actually faces the capillaries or blood. This is the basal surface. So all these luminal epithelial cells, they have got two surfaces. One is the apical surface or the luminal surface, another is the basolateral surface or the basal surface. So what happens when we take some food or anything, actually the transport process, it occurs through the lumen across this luminal surface of the cells. This cell membrane will take part into the transport. Okay, the energy that will be utilized, it is not at this luminal surface from where actually the absorption is taking part. So where it is now utilized? It is seen that in the basolateral surface, what happens, the energy is utilized via this sodium potassium ATPase pump. Okay, so what happens, suppose I have taken the ORS which contains the sodium and glucose. The best example is SGLT or sodium dependent glucose transporters. Okay, so what happens, when the glucose and sodium is coming into the gut, the entry of glucose, it is coupled by the sodium. Okay, so how it happens at the basolateral surface, actually the sodium potassium ATPase pump is acting. It is utilizing energy, it is utilizing ATP. Okay, and as a result of the sodium potassium ATPase pump, we know that its function is to expel 3 sodium outside the cell. So intracellularly the sodium concentration will go down. Okay, and along the concentration gradient by passive way, this sodium will enter into the cell. Okay, the concentration gradient is created by the sodium potassium ATPase pump, which is acting as the basolateral part. But from the surface where actually this transport is happening, here no energy is utilized. Here there is passive transport along the concentration gradient of sodium, and when the sodium will enter, it will take the glucose with it. That's why it is known as the sodium dependent glucose transport or SGLT. Okay, so this secondary active transport, it is an active transport. Here the energy is required, but it is not at the surface where actually the transport is happening. Okay, that is the example. They can be co-transporters where direction of ion movement is same. Now it can be the antipode or exchangers where direction of ion movement is opposite. So it is an active transport, but it is secondary. The best example you will see in gut and the renal tubule. where there is sodium dependent glucose transport happens. These are the references, and thank you.